Yeah. Wait, are you gonna
Oh, we're live.
introduce or should I go?
Hello and welcome to today's Wolfram live stream. Today we have Willem Nielsen talking about robustness for um, cellular automata, adaptive systems. So kind of biological medicine or CAs. That's kind of the theme. Willem, go ahead.
Yeah, thanks, Dugan, and thanks, guys, for coming. Um, yeah, so like Dugan said, I'm just gonna be trying to I'm gonna do some live coding to look at try to get some insight into how we can better emulate the robustness of biological systems. So just to give you some background, so we're working with the Broad Institute, and they have this nice paper that looks at it's a genome-wide atlas of human cell morphology. So what that means is they just did a bunch of single gene perturbations and took a bunch of images of biological cells and stained them. So these different pictures, yeah. So like these different pictures show stainings of different organelles. So like the Golgi and cell membrane, the nucleus, etc. And so what's cool is they can tag these cells so that when they image them, they know which genes in the cells were perturbed. Um, and so they're kind of trying to create a map between gene knockout perturbations and image features. So to be more specific, they took like thousands of different image features. So for example, cell area or like granularity, and then they try to map them to different genes. So what I'm kind of trying to do with this data set, though, is try to connect it to our recent, biological evolution model, and work in biomedicine. So, and by the way, Dugan, feel free to stop me and ask questions or add on. Um, but so what this is showing here is just a bunch of different, a distribution of different metrics based on the images. So, for example, this is the area of the cell in response to different single gene perturbations. Um, and what's kind of what was striking to me looking at this data, um, and I'm going to collaborate further with to understand it better. But the thing that jumps out at me is just how like kind of smooth the distributions are, which is a little bit different to what we've seen with our models. And so I kind of am looking to you know make sense of that. So, to be more specific, this is our recent model of biomedicine, the CA that kind of the model CA that we used for the post. And by the way, Brian too, I should say, or Max or Luke, if you guys have questions, stop me. But yeah, so this is the distribution of the length of the cellular automata based on single cell perturbations.
Okay,
Um,
so yeah, let's
so
just
what's different?
talk about what's going on here. So you have this. How many states is this? A three-state Turing cellular automata?
Yeah. So this particular one is k equals 4 and r and
Okay,
radius of 1.
so it has three colors and then a fourth color for white.
Mhm.
Yeah. Okay. And then it's being seeded by a single dot up top, and then the CA develops from there. And you chose this. You did a search through the space of cellular automata to get ones that end at a finite length, and then you started testing them for to see how they behave under random single point attacks. So those are where the arrows are changing one of the values in the evolution, and then let's see what the cascade effects are downstream from that perturbation. And then this is robust in the sense that it uh, doesn't get too crazy, except at the last one where like you had one point mutating, uh, causing a very drastic cascade effect from the original form. Is that a
Yep.
good summary?
Beautifully said.
All right.
Um, Yeah, that's a really good background. Um, and that's exactly right. So, and the reason we chose, by the way, uh, actually, I don't know, it's not important, but the reason we chose k equals 4 is it's just like the fastest to evolve that we found, but we haven't looked much at the higher ordered, like k equals 5 and above, which is part of what I'm going to do here. But yeah, yeah, a lot of stuff is not known about like and this is kind of why I'm doing this is like what's the robustness, what's the relationship between different rule spaces and robustness. Yeah, so these are some other distributions that we got from a similar algorithm of evolving cellular automata under perturbations to reach a longer and longer halting length, as Dugan mentioned. And so what's again, a lot of times, sometimes there is like some smoother distributions, which is kind of closer to what we kind of want to see in terms of comparing with what we see in the morphology data set. But a lot of them still kind of have this spike or kind of like weird discrete, kind of their discreteness is still shining through a little bit. Um,
Yeah, some of
and
these
it
look
seems
like
like,
just bimodal distributions. You got two peaks, two bell curves stacked on top of each other,
yeah,
like two sweet
yeah. So it
spots.
seems, and it seems like cells don't really have that. It seems like they have kind of enough parts where when you perturb them, their kind of features are smoothly just change in a smooth manner, kind of. Um, so yeah, so the question is, how can we kind of emulate that? So the first thing to kind of look at, I think, is just looking for patterns that run for longer steps, and so they kind of have more parts, so you can make smaller perturbations to them. So to give you some background, a human cell has 20,000 genes. So when you're changing a single genome, it's much smaller. A change to a single gene, I should say, is much smaller than a change to a single cell in this pattern. So this pattern has about 4,000 different possible perturbations and only a few thousand cells, I think. So basically, the percentage change of this perturbation is much larger than the one in the data set. So we want to get larger patterns.
Talking about the
uh,
space of rules for the poor color cellular automata.
Not the space of rules, the just literally the number of colored, what we call body cells, or like
Oh,
cells
I
that are
see.
in the non-zero range.
The size of the
So just
phenotype
literally
then.
bigger patterns. Yep. So, okay, so this is an example of that. These happen to be k equals 5, but the important thing about them is they're much larger, and so they kind of have bigger parts, or sorry, more kind of components that make up the way they achieve their objective of living some finite length. Um, and by the way, it's kind of interesting that these guys look, to me at least, they look more like biologic. Um, so I
Yeah.
think it's kind of getting at the level of complexity that we're used to seeing in biology. Yeah, so like for this pattern here, it's not quite. To me, it seems like it's a little almost too simple compared to what we're used to seeing in biology, as opposed to something like this, which seems more like lifelike, at least to my eye. Um,
Yeah, it looks like mold
yeah,
or dendrites or like a flowering plant spilling over.
yeah, for
Um,
sure. Um,
some of those at the bottom have lot more like rigid structure, I guess. Kind of looks like
yeah,
things on a shell. Oh, oh, that's
yeah. So
interesting.
like a lot of these are, yeah. So like for example, this one I would say is seems more, and like Stephen kind of was looking at this with our Rulli ensemble project was, um, it kind of feels less biological and more like it was engineered because it's kind of like I don't know, there's more geometry and straight lines as opposed to
It,
something
it
like,
looks like a
this one.
sword you would find in like a Nintendo game.
Yeah. Um, yeah,
There's
like this
one
guy is having
below
a
on the
team.
bottom row that had very interesting features. The one that
Which one?
this one, yeah. So that triangle thing up top like starts replicating itself. It looks like
Hmm.
um, you see that motif happening a lot.
Yeah.
It's very interesting.
Um, yeah, so I mean, one kind of thing that's also interesting is that these are symmetric. So, and they tend to have, as you're kind of pointing out, they tend to have more, I would say, repetition in their pattern, um, than asymmetric rules. And
Yeah,
we can actually look at, uh, so like here, I'll just plot the asymmetric ones, and you can kind of see that it looks a little bit less biological because there's kind of, well, first of all, I think symmetry is really common. So when our eye sees symmetry, it kind of think, it seems more,
we like
like
it.
what,
Yeah, yeah.
let me, yeah, so it's this, and then just, yeah, so these are asymmetric, the same setup, but just asymmetric. And to me, at least, they look less biological. They look kind of more alien almost.
Hmm.
Um, and they don't have as much like repetition in their structure. Um,
Yeah, I mean, just thinking about it in like biology, like even slime molds have like spores that are mushrooms that are like radially symmetric. I guess that's a function
yeah,
of the way you want to construct something large using cells. You want to make something out of like a bunch of cells, so it's like you tell one cell to like go next to the other and like go next to the other, and you sort of have like a construction method that has some sort of symmetry inherent to the system.
yeah.
But like a
I
slime
mean, that's a
mold
really,
would have asymmetry based on whatever type of environment it's trying to grow into.
yeah. I definitely don't think it's always symmetric. Like, yeah, like you're saying, with slime mold. Um, like for example, proteins come to mind where they're just a lot of them are just, a complete just jumble.
You're right. You're
It
right.
doesn't
Yeah.
seem to
So
have
the structure of a
structure.
protein is sort of you're putting amino acids in a chain, and then that's like folding and crunching up ad hoc based on the electrostatic forces between the alpha sheets
Right.
and the beta sheets, or the alpha helices and the beta sheets. But yeah,
Yeah. I mean, definitely with like, morphogenesis of like higher level organisms, it's a very symmetry is like almost ubiquitous. Um,
right, right.
and
So for like
why
body
that is.
type of the embodied organisms tend to be symmetric, I guess, because that's easier to construct macroscopically.
I can't prevent myself from jumping in.
Please.
The only things that are symmetric are things that walk. And if you want to have a direction of motion on a surface with gravity, bilateral symmetry is a good trick. The plant kingdom tends not to have bilateral symmetry because they're not going anywhere.
Very good point. Yeah.
Yeah.
Interesting.
But plants do have symmetry.
The tree right out the window here that kind of has like broken radial symmetry.
Hmm.
And there's another things that makes the morphology of trees, for instance, and I think it's true for most plants, is the locus of growth um, is only at the edges.
Right.
Right. That the stuff on the inside is really not changing very much.
Hmm.
But I didn't really want to jump in when you were in the middle of the presentation, so um, when
I
you're
was
at the end, we could pick this up maybe.
totally welcome for
Yeah,
a discussion.
Okay,
yeah,
no, because a comment that I wanted to make is if you go back to the original graphs that you got from the Broad Institute, um,
oh yeah.
and it's funny though, because I know that my kind of reflex reaction, can you get to the graphs place here?
Um, Sorry, what do you mean?
There were some histograms here.
Oh, here we go.
Yeah, here. You look at these histograms, and it's funny though because most of them are saying law of large numbers because it looks vaguely Gaussian,
Mhm.
right? With the exception being the second one in the second to last row. You know, that one's looking like, the decaying exponential pattern.
Mhm.
And it's kind of like one of the things is we were doing a very similar session with a different thing, and Willem, the live coding you were doing was playing Game of Life games,
Mhm.
and the graphs that you were getting, it wouldn't surprise me if you got similar graphs.
Yeah,
That um, I think it could be the case that if you
One moment, folks.
many of them fall into either the Gaussian kind of pattern, and many others fall into the decaying exponential pattern. And it's often the case when I was running into graphs like the ones that you were showing. Um, it was kind of like it um, there isn't enough there yet for it to be for the law of large numbers to kick in. Right. And in particular, by the way, if you go back to the histograms on your side, um, the bimodal one was something that I saw with a lot of CA stuff because the first the peaks sort of correspond to one peak is the average, but every now and then you get an arpentomino, and the arpentomino leads to a second peak further down.
Mhm.
Right. There's kind of like it's even when we were saying with the permuting the game of life things in the last session that we did. Um, Most of what we did made very little change, but some of what we did had a very dramatic change. And
Mhm.
this may be a question for the Broad people. Is it the case that you would think biologically, you would think that in biology, the two modes would be do very little or make a small change? Where in CA, there's a third category, which is cause something big to happen.
Right.
Right. And, I don't know whether or not the cause something big to happen is a pattern that you'd see if you're working with actual cells.
Right. And that's what I'm wondering is because, like, I mean, to me, it seems like disease is indeed at least somewhat more like a exponential. Like, um, I don't know. It seems like when things go wrong, they can go really wrong. Like, you know, I don't know, with cancer, for example, like, the if you just
But,
look
but,
at the
but
number of cancer cells over time, it can grow really rapidly. But it
no,
doesn't seem to show up in their data, which is kind of
it's funny that what I was working on when Dugan pinged me to join this meeting is I was just starting to see how the sets of metabolic reactions in E. coli connect to each other. Um, because you could be looking at genes, but there are various classes of genes. One class of genes is the genes that make proteins, right? The genes that make proteins, what they're doing for a living is they're implementing control knobs for a big network of metabolic reactions. And it would almost be, and this may be again a question for the biologists in this, um, if you're looking at the gene knockouts, they probably, for a lot of them, that may be a lot, it may only be 30%, could say, what was that gene doing for a living? and sort of saying if it's replication class gene, how does that change the morphologies relative to a metabolism control class gene, relative to a, cell membrane class gene?
Yeah, I mean, I guess if if there's a gene mutation in a cell, it could not have very many effects, or it could, change the if you make a change to the protein to the gene which codes for like an ATP synthase protein, then like you're gonna have a bad time.
Well, no, actually, when we were mentioning cancer, there's been various theories about what cancer is about, but most of them, at some level, say what the disease has done is made cells that don't know that they shouldn't be reproducing as fast as they can. As the growth regulation mechanism gets knocked out. And actually, I don't know what the current state of affairs was for a long time. The consensus theory was the way it got knocked out is that there was a gene got zapped and that did it. There was a more recent theory that I don't know if it got to the point of consensus of it's not genes actually, it's interlocking metabolic reactions or something like that.
Right, right. I mean, Michael Levin kind of has the viewpoint of cancer being a breakdown in intercellular communication of a part of a tissue with the surrounding tissue. It goes rogue,
Right.
essentially.
And it's funny that I know nothing about this, but the wild-ass guess I had is the thing that's broken should be something broken at the decade's time scale, where genetic stuff is at the millennia time scale and pure metabolic stuff is at the day time scale.
Mm.
Right. So the question is what kind of regulatory mechanisms do we have that, because what it is it's basically it's breaking cellular differentiation. Is, somehow, the liver cell forgets that it's a liver cell and it turns into basically an early stem cell, which just grows up a storm because
Yeah.
it doesn't know to be polite. So maybe getting back to the things that you're talking about. Do you actually think that the simple CA is at the right scale for doing this kind of experiment?
It certainly is
Yeah, I mean,
analogous.
I think short answer is yes. I think the ones we're looking at are not quite complex enough to, get perfectly similar behavior, but like the core features of what's going on, I think so. So maybe
Yeah, we can keep going.
yeah, so maybe it's good to keep going now. Yeah, so okay, so let's see what happens if we look at, so first of all, let's just see what happens if we perturb these larger patterns and see how robust they are. Okay, so I need, okay, so this is just perturbing a random sample of them. So that kind of gives you a sense. These are kind of different. So let's look at just like the largest three patterns. So that's, yeah, so this is,
Zoom. Goodness.
fragile. So for example, this cell, this one has, I think like 100,000 possible perturbations, and we're only doing one, and it completely changes the entire pattern. So this is very, extremely fragile, not like cells at all in this particular case. But part of that, I think, it really just depends on the pattern. But, we can also look at just the distributions. So let's try to just, so let's just look at the first three. So this is a random sample of, 100 perturbations to the three largest CAs of k equals 5 symmetric. Yeah, I mean, these are pretty not nice. Maybe if we do some more. Yeah, so we're kind of getting those similar kind of spikes that we want. Yeah, so let's try to see. I think part of it is the symmetry makes it fragile for some reason. So let's just try this exact thing, but with asymmetric ones. I think this should work. Okay, so these ones are almost even worse. Interesting. Yeah, so by the way, these like when it goes to 0, it means it ran past the length of the cutoff. So that's what that huge spike is.
I see. So that means a lot of them went long, got perturbed, and then just started going crazy, cancerous, essentially.
Yep. Okay, maybe if I do, kind of just want to see some of these perturbations.
So the comparison that you're making is between a closed loop system and an open loop system. Is there anything CA-like that we could do that in some way closes the loop?
Say more, what do you mean?
Everything in life at all levels has some kind of homeostasis. You know, instances of simple programs that are trying to stick around the set point.
Yeah, so that's really what this guy is. Because when we evolve it, we make perturbations to its pattern, and then we define its fitness to only be the minimum of the perturbation. So if it's really fragile and it just explodes into cancer when it changes one single cell, then it won't be able to survive, kind of. And the way it kind of does it is it's like an attractor. So it just kind of all roads lead to this purple. And then once you get to purple, you get to white, if that makes sense. Yeah. But I'm wondering if, so kind of like a practical thing is to actually evolve to these patterns. It's very computationally intensive because in doing perturbations, you double, triple, quadruple the number of simulations of the pattern that you have to do. So it's kind of challenging to do, and I'm trying to find robust ones that are larger to match this kind of the size of the genome. But so I'm wondering if what happens if we just increase the number of parts of the system? Is it possible that in and of itself will make them more robust and like kind of have the feedback loops emerge without having to explicitly, run the simulation? You know, maybe it's kind of optimistic, but I don't know. Does that make sense?
It's funny though, because it's not really the size, it's the ratio of the ratio of silent mutations to the ratio of non of mutations that push things to a different place. Right? Because
Hmm.
the thing is they're doing those experiments with human cells, right?
Mhm.
And don't we think that in any given cell, there's really a small percentage of DNA that's actually active at that point in
Mhm.
the overall evolution of things?
Yes, I agree.
Okay, to the extent that that's true, they're going to be able to hit it a lot more often with no effect than you are.
Yeah. Um, so one thing we can do to kind of look at that is, so right now I'm perturbing, and to your point, I'm perturbing a cell that is indeed getting used, but not all genes are really getting used. Um, there's some redundancy and you know, so we can actually look at that by just looking at what happens if we perturb the rules. And some of the rules, I suspect, um, aren't actually getting used in this pattern. So we can see, so let's look at that. So, okay, so we just basically want to do this. And so let's just take first one of this. And then, so I'm just going to make a single change to the genome and then, or, you know, the model genome, and it should sometimes have no effect, just like we see in cells. Let's see. Okay, that's not encouraging.
Maybe get a different. Oh, I see. You got K5 radius 1, set A, the first and the first.
Yeah, so let's see
So
what
is
happens if
that
we just do. Huh.
I mean, is should be using like maybe a different rule.
Well, let's see. I don't know that it's possible that this is just not working. Yeah, so I don't know what happened with that, but that rule is not right. Let's see. Maybe just choosing a different one. Oh, that one
Because you're using
had a
you're
fitness of
picking one at
1.
random, that bottom one. So if you ran that
Yeah.
again, you get something else.
Uh, yeah, I would. I didn't see it, but here we go. So,
Okay.
okay, so this is,
All right. So
yeah, so
yeah,
Brian, this
that
is,
yeah, it
oops,
runs
never
for like
mind.
500.
Okay, yeah, so,
And this is a random change to the rule set, right?
yep, so now instead of changing a cell, we just changed a rule. But, so it seems like these ones are still very sensitive to changes in the rule.
Mm-hmm.
And at least in this run, let's try again. Yeah, so, I mean, what was the original pattern? Yeah, so that's the original, and these are all the randomly gene mutated. so to speak. Yeah, but so I guess for this one, so okay, so this is a k equals 5 genome. So that means that there's 5 to the 3. So there's 125 rules. Now, how do we see which ones are active? I think it's plot rule. Nope. Oh, maybe it worked. No. Yeah, I know. I gotta fix that. But yeah, it'll be interesting to see. Because I think it's a good point, like how much of our rules, how much of the rules are getting used, and how much are the genes getting used, and is that similar?
Yeah.
Okay, but let's look at some more higher colors and just kind of keep going. So this, we're just going to evolve a k equals 6.
So this is evolving for what type of metric? I guess that was evolving a rule set
So
to
this is just,
find one that like has a good behavior.
it's just evolving for maximum length up to
Okay.
1,000.
I see.
Yeah, so these are the ones that we got. I like the blue.
Yeah.
Okay, so then let's see what happens if we perturb, say, this fourth one.
So 704.
Yeah. No. There. Okay. So at least, so yeah, so this is what I was kind of hoping. It seems like to me this one's a little more
It's fairly
robust than the other one. Let's see if we do it again. Yeah, see, this one isn't bad. And this is kind of what I've seen before, which is, I think more colors means a little bit smoother variation. Let's look at the histograms.
Actually, to make sure we're on the same page, what exactly do you mean by robust?
Yeah, so I just mean that when it changes, sorry, when it gets perturbed, the lifetime or the length doesn't change much. Is that what you had in mind, or?
Yeah, it doesn't change that much when attacked by a single point
Well,
mutation.
Yeah, no, this is the thing that I was wondering is robust could be defined as percentage that stays within a bound, or robust could be defined as minimizing the ones that are vastly out of range. Right? You could define a definition of robust as 85% of them don't change, but 15% of them could be anywhere. Right. Or you could define robust,
Yes.
you know, 99% of them are within certain error bars.
Yeah. Yeah, that's a fair point, I think.
So which of the two are you going for now?
I mean, I'm just
Yeah, wait, so what does
just
that what does
looking
this
to kind
plot
of
mean?
like, okay, so this is just 1,000 random perturbations to this rule up here,
Uh-huh.
and these are the resulting lifetimes.
So there's a peak at 700 at the original lifetime length.
Yeah.
Right. Just
And then
wider aesthetic.
there are a lot of them
Why
that
did you
make everything blow up, I guess.
you're putting the ones that really blow up on the left rather than on the right?
Yeah, I can change it easy now. Yeah, it's just weird because I'm just gonna leave it because it's it's running to 2000, but there's actually none that get that far. So if I were to replace it with something large,
Oh, wait a minute. Wait. So
wait, okay, wait, we should just, to avoid confusion, do this and then do this, or maybe, okay, yeah, so actually that wasn't quite right what I had. Yeah, but this is where these are now the whatever, infinite patterns.
Mhm.
But yeah, so again, this one is an interesting distribution. Let's see what happens if we do more. Yeah, same. So it's kind of weird because it's like on the left side, it's one distribution, on the right, it's another.
Oh, wait a minute. Wait, you changed the other there. You didn't run them 2,000 steps or 5,000. Well, if you
Okay,
change
so
that other 2,000
this,
to 5,000, do we get a different plot? Yeah, right there, I guess.
this one,
Yeah.
no, because this is just the, it's just finding the possible perturbations.
Oh, I see.
This should be the exact same. So let's see what happens with the other. So maybe this one is a little bit smaller. Yeah, I mean, at least one thing we can kind of conclude is doesn't seem to be, it varies a lot rule to rule, it seems. Let's see what happens if I just do automatic binning. Yeah, so this one is like just one giant spike.
Hmm.
We should also do to see the peak. Yeah, so that's that one.
Gonna
Again,
say,
not very.
you put a log in there.
Oh yeah, that's a good idea. Let's see.
Hmm.
Yeah, kind of power law-y, although yeah, we would need to see the bins in log too to see that.
It is, look, can you just do 10 times the number of goes? Or would that,
That's a little scary, but
that
I'll
would
do
that cause
double.
this meeting to last until the evening?
Yeah, it is smoothing out a little bit. Let's try. Oh, sorry, this is the wrong number to be changing. That's why it's so fast.
Oh,
Yeah, so it should be this guy. Or no, this one. I mean.
hmm. Yeah, it's a bit smoother.
But the holes
Uh,
didn't get more numerous, did they?
hmm.
You know, it's like it looks very, very smooth, except it's like the first hole had its stuff moved over a bit.
Yeah, maybe I should keep the old one for comparison. Hmm. Yeah, I don't know. Pretty similar. Okay, So maybe I'll try a few more colors and then see if, and then we'll wrap up. Okay,
thing.
so let's run this guy. See if we get anything. Okay, so this is k equals 7. I should change the seed. Okay, I guess we can try this one. Fourth one. See how it looks. Yeah, it's very lots of variation there.
It also seems very
Let's see how
modular, that one.
histogram. Yeah, good point. Although, is this the same rule? God, this is so annoying. Here, let's do this.
Mm.
Let's see, modularity. I don't know. Kind of. Okay, let's try to do the histogram. Oops. Histogram. 4. This is V7. No. Huh. What happened? Oh. Oh, were these k equals 6? Okay, let's try this one.
Oh, it's being wrapped.
Yeah. Should probably make this like this. That doesn't happen. Or,
Okay.
yeah. I mean,
So
a lot of
it
these seem like power.
sends out a glider, essentially.
Yeah. A lot of these seem like power law, like, which is interesting. Okay.
If you flip pennies and flip pennies for as long as you get heads, the run length will be a power law,
Yeah.
right? So,
Yeah, that's
right. So power law distribution is it's kind of like a run length of a random choice.
yeah.
So the question
Sort of,
is in the experiment that you're doing, what's the random choice that we're seeing the run length of?
yeah, I guess it's kind of like the whether or not it grows, like increases in width or decreases, kind of.
Well,
But yeah, this.
is there any way to add another dimension of the graph and say how far down in the evolution the perturbation happened?
What do you mean?
Maybe I was misunderstanding your first graph. How are you? I thought that if you go back to like one of the earliest images, I thought you were just nuking a cell at some number of steps down in the evolution.
Yeah.
Right. but is there a way of keeping track of how many steps down the, it's kind of a scatter plot between
I
how
see.
many
Okay.
down and how
Okay, yeah.
big the run make the changes?
Okay, let's try it. So we basically want to do this. Uh-oh. It crashed. That sucks. Dang. All right, I'm back. Share my screen. Yeah, okay, so let's try to, and I just need to run the code notebook.
Kernel crashed.
Yep, just or the application.
I see.
Okay, so we need to get this guy again. I think I can just run this. And then we can write this code. So we want this, but we want to do it at successive steps. So it's going to be like this, random table. Let's just try that and then see if it's working.
Personally, I blame the servers. Oh, look at that.
That did not look right. Oh, okay, because it's so early on in the pattern. So maybe if we do at the 200th step. Oh no, that's doing 1 to 200. So is that right? No, it's this. Okay. But Dugan, you have to go, so
Yeah.
maybe we could do this another time. But I don't know. I thought this was interesting, and thanks you guys for the questions, comments. Because I really
I definitely
yeah.
appreciate it. Well,
Mhm.
thanks for showing us around. This it's fun looking at these CAs and sort of seeing what they do when they get perturbed and trying to find good ones, gems among them. And yeah. All right. I have to get going now. Thank